No. So hello everyone, welcome to this seminar. Today we will listen to Louise, who will talk about her work as a postdoc here at the LVA. Louise, la parole.
Hi everyone, thank you to be here and to listen to me while I talk about my postdoctoral research. So today, as he said, I will talk about acoustic monitoring of frog choruses using a network of 5 spherical arrays. And I'm doing this work with Quentin Leclerc, Pierre Lecomte, and Jérémy Rouche with the LabEx Celia. So I will start by giving you the context of this study. Then we will see the methods and the results that we could have. So first, for the context, we have to know that amphibians are among the most vulnerable species to the climate change. And this is due to 3 different things. So for the first is that the frogs are really sensitive to small changes in humidity and temperature. Also, the use of pesticides is really dangerous for them because they have a really small, thin skin. And last of them, the fragmentation of habitats are really disastrous because because of the transport infrastructures. So that makes them the really endangered species, and so that's why they are fully protected by the Nature Conservation Act of July 10, 1976. So that means that you can't remove their eggs, you can't destroy, mutilate, or capture them, or trade those species. So what we will talk about during this seminar is more specifically the vocalization of amphibians, and 3 of them, because that's the one that I studied. So the 3 of them are the green tree frog, so rainette verte in French, the marsh frog, grenouille rieuse, and midwife toad, les alites accoucheurs. I will make you listen to those 3 vocalizations because they are really different, and I think it's nice to hear them. So the first one is the green tree frog. This is the marsh frog. And here it's interesting because you can hear 2 types of vocalization. One is like the laughing call, which is the one that they use to attract mates. And the other one is the wah call, which they use to fight for their territories with other males. And the last one, but not least of those species, is the midwife toad. That's the one I will talk about during the rest of this seminar. So it's this one. So this one have a really specific call. And also you can hear that in this particular case, you can listen to 2 males, because if each male have one frequency call at one really particular frequency. And what you can also tell is that their call are really single short tonal calls of really short duration. So that's what we will see later. What I will talk about is the choruses. So what happens in a lot of insects, but also in aneurons, is that frogs and insects gather together, so aggregate in order to vocalize and attract mates. But not also for that. It's also because they want to be they want to lower the predation risk and to attract females that are further away, because if they vocalize just individually, they will not be loud enough to be heard by females. So that's why they do that. What have been done in research earlier is a lot about the timing of their calls and like the intensity of their calls, but not about the spatial dynamics. So that's what we will talk about. the question we try to answer during this postdoc is what is the spatial and temporal organization of male frogs, and in particular midwife toads. So what we can tell is that passive acoustic monitoring is an emergent issue in bioacoustic community because it is a non-invasive way to study species and to study communication between individuals and also to assess populations. So the usual way to do that is to use one single microphone. So you just put one microphone like in a forest, for example, and then you can tell by the registering of this microphone the diversity of the biodiversity. But it's not enough if you want to have the localization of individuals. So what people do usually is to use an array of microphones. So that's like microphones where you know the position of them so that you can know the direction of arrival of the sources. But it's still not enough if you want to have the 3D position exactly of the species. So what we will use in this presentation is an array network. So you have several arrays, and with that you can localize really precisely in 3D the position of individuals. So now I will present the method that we used to do that. We use a network of 5 spherical arrays of microphones, which are Zilia arrays. So you have 19 microphones per array that are baffled around it. And the size of each array is like 5 centimeter radius. So what we will do is that we will deploy this network of array in the field. And then we will do sound event detection and then locate the sources. So that's 3 main parts in the method. And I will describe them more precisely now. So to describe the method, I will use an example so that it can talk by itself. So this is an example of experiment that we did in Parc des Alites in Villeurbanne in June, because that's the breeding season. It begins like in April and it ends in August for midwife toads. And so here is the video. So as you can see, it's at night because those species are vocalizing from like 9 p.m. to 4 a.m. And also we had kind of we had to put our arrays in some specific locations because we couldn't enter in the ponds area. So here it's the 2D sky view from the site we were in, so in Parc des Alites. And you can see that we put the arrays in this path here. And the photo here is taken from this point of view, from this row. Also, what you can see, so the ponds are around 5 meter length. And in between those ponds, you have stone walls here and over here that are in the right in the photo. And what's important is that we know that Alitopstetricans like to hide in those stone walls and also in bushes. So maybe that's where we will find them. So once we deployed the material, we try to have the same orientation of each array in the field so that it will be easier to know the correct location of each array. So here the position of the array are oriented towards the X direction, and each of them is like this. So what we do to geometrically calibrate the system is that we will use acoustic signals. So we will do 14 hand claps around the setup. And then we will use the time difference of arrivals of those hand claps to each of the microphones. And to estimate that, we will use generalized cross correlation on each of microphone signals that we have. So here you have each color is for one array, and you can see one hand clap that is seen on every array, and it's quite easy to see the difference of arrival. So with that, we will, once we have the time difference of arrival, we will try to minimize this cost function. So tau, as we said, is the time difference of arrival that we experimentally had. T is the simulated one. And O are the errors that we can have with the calcul of TDOA. So we do that by majorization minimization theory. So once we have that, we have the position of every array. We will try to find if the orientation of each array is good, because in the field it's quite complicated to position the arrays. Sometimes it can be oriented in a specific way. You don't have flat floor, so it's not easy. So we will correct the orientation using the direction of arrival of the claps that we did, and try to minimize another cost function involving the delta tau here. So the difference between the direction of arrival that we experimentally have with beamforming and the one that would be the one for the source S0 here. So this is working quite well, but we still have 2 centimeter error in standard deviation on the microphonic positions for if we have initial errors of like 20 centimeters at the beginning. So yeah, we still are aware that this method is not perfect because we don't have the sources time synchronized with the microphone, so it's making it more complicated. So now we'll talk about the sound event detection, because now we have the position of the arrays and we have to know the detection that we have. So I will make you listen to the extracts that we will analyze later. So this is a 10-second signal, and here you can hear that there are a lot of allitopsetricon. There are like maybe, I don't know, we hear a lot of them. And we will look at the spectrogram of the one of the array, which is this one. And here you can see more specifically that individuals are calling between 1,200 and 1,600. Yeah. So this is quite not a wide band in frequency. And also you can see that the duration of each call is really small. So we will be able to extract the calls and then to localize each call individually. So that's what we try to do. And to do that, we will put a threshold on the spectrogram, so to have only high amplitude vocalization. And then we will do clustering between those high amplitude pixels, so that we will have each vocalization, which will be like one cluster. So the aim of this process is to have one individual equals one color here. So you can quite clearly see in the first spectrogram that there's one individual here at 1,350 Hertz, which is quite clear here. But the other one, it's not that clear, except maybe this one. So visually, you can already tell the number of individual, and it's around like 5 here. Now, if we use our methods to do clustering and with the pixels, we can see that the individual division here in colors tells us that we have like maybe 17 individuals. So obviously it's not true, but we have some errors due to merged pixels and stuff like this. But still, it gives us an idea of who is calling when. So once we have detected those calls, we will do localization for each of the events that we have. So one event equals one call. The hypothesis that we have for each array is that the sources are in the far field compared to the array, so we can tell that we have a plane wave arriving from azimuth and elevation precisely. We also know that we have the 19 microphones of each array, which are precisely known in position. So we know the positions of the array, of the microphones on each array. And also, as we have a spherical array, we can express the field in terms of spherical harmonic. So we will use spherical harmonic beamforming. And this is the result for one of the array, for example. The idea is that the call will be pointed with the little cross here. So it's the highest amplitude point on the grid. And once we have the direction of arrival of each sources, so of each call on each array, we will be able to plot that. So for example, for the Z0, this array, we can see the clusters of points here with different colors, which corresponds exactly to the colors in the spectrogram. So for example, the green individual here that calls like 15 times will have 15 points with the green cross here. And as we can see, all the individuals are quite at the same spot, but still we can see clusters. So we do that for the 5 arrays, which will give you the 5 different projections here. And once we have that, we will try to pass at the global scale, so to have the position in 3D of the individuals. So here it's still the sky view, and you see the 5 arrays here in white and black. And what we'll do is that for each event, we will trace lines between the center of each array and the respective DOA, the direction of arrival of the source that we detected. So that's one of the examples that we have. And for example, here you can already see that Z4 is not seeing the same source as the other ones. Then for each pair of array, we will minimize the distance between those 2 lines, for example. And this minimization will give us the triangle here. So we do that for every couple of lines, and that's the results. After that, we will give a radius r of like less than 1 meter, and each point that we plotted that are not in this radius will be excluded. So, for example, here this array will be excluded from the thing. And the last thing we do is that we keep all of those lines and we try to find the points that minimize the distance between all of them. And it will be the point P. Okay, so we do that for every event that we detected, and that will give us those results, those kind of results. So it's still the same spectrogram and still the same 10 seconds. So now you have seen it a lot. And so here, the first individual seen as green here is located, you see here around the stone wall, like it's just next to the stone wall. But you still see that there's a cluster and not 1 point. The second individual is located here, the third one, the fourth one. So here we can see that the green individual is further away from the arrays and that the other one are gathered together. So that's with all the sources. And here is an example of detection file. That's what we wanted to do at the beginning, to have each event with the number of events, the number of individual, the time of each event, the frequency, intensity, and position of sources. Now what we can do is that as we are sure that every color is one individual, we can take the median of the positions of the cluster of points for each individual, which will give us this kind of cartography, so that it's clearer and more easy to see it. So now that we have done that, we can conclude. And so the question was, what is the spatio-temporal organization of male frogs in choruses? So we can answer in two ways. First, for the chorus, we can tell that here obstetricans are vocalizing around the stone wall, and that they are still really close one to each other, except the alit with the lower frequency voice. And now for the localization method, we did experiments in an anechoic chamber at first with a simulated source to check the method. And then we did it in several fields, so with alit obstetricans and marsh frogs. But this system can also be used to study any species, as long as it's in a frequency range that is okay for the system. And now, in terms of prospect, what we want to do is to have a better geometrical calibration, so that we can use other methods in the localization in 3D. Because here we use the geometrical method, but we want also to use like TDOA or Bayesian methods, which we can't use because of the little error in the calibration. So if we don't have exact positions of the microphone, it's difficult to use other methods. So what we are doing now with Valentin Minot in LVA is to have sources that are time synchronized with the microphone signals, so that we will have less errors in the calibration methods. Thank you for your attention.
Thank you very much for your presentation. We have some time for some questions. Do you have any question, maybe? Yes.
I say like when you look in the separate of the microphone, but when you off the straight direction, the result you want to show in the 2D, you have more information.
Yeah, because it's hard to. I could have put 2D in XY and 2D in Z, for example. Yeah, but I didn't do it. But because the important thing for me is to see, like in 2D, how it happens. Okay, you're right in the sense that we have a 3D network, so and we are able to locate in 3D. So we would like to study species that are in 3D, but here it's frogs and they are on the ground, so it's not really interesting to see the height. But we can see it anyways, and we can see it's on the floor.
Thank you for the presentation. I just want to ask, how did you choose the ranging on the different, right?
Yeah. So the best, if you have spherical arrays like this, is to place them in the most spherical way, but we can't because we have a human size, and also we have special conditions in the field. For example, here we couldn't go in the ponds, we just could go in the path. So that's a bad way to put them, because they are like in a line. And so this direction here is not well seen, because as you have seen here, the direction of arrivals are really close one to each other. And so we have an angular error, which is bad if we have aligned arrays like this. So we try as we can to not do that, but sometimes we can't do something else. Yeah. Yeah.
Yeah, maybe I didn't understand everything that you meant. If you try to assume that the inherent to the method of localization is the assumption that the sound propagates as in the free field.
Yeah.
Without reflection.
Yes.
So what, in your opinion, would be the effect of the ground environment regarding such?
Well, for the direction of arrival, it can have some. We can have some problems with having the right direction of arrivals. Yeah. Yeah. But here, I think in this special case, I think it's quite okay because we have 5 arrays and a lot of different events that we can study. So overall, I think it's quite right. But yeah, we are assuming a monopolar source in a free field. And actually, there have been some studies of the directivity of the vocalization in frogs, but here we assume that it's monopolar sources.
So we have also a question from the YouTube live from Jean-Pierre Coyet, who asked that the measurements seems to have been done in urban area.
Yeah.
What about the parasite sources?
So the experiment have been done, yeah, in an urban area, but it's at night. So we have sometimes cars passing by, but really not a lot. And also it's not at the same frequency bands. You can have insects also, but insects are in higher frequency. So here it's not really, if you see the spectrogram, it's quite clear. It's not really affected by other sources. We also have like dogs and human passing by, but I mean, it was really okay. We also did some experiments in a more natural environment, but it was not with Alitopstetricans, so that's the species I wanted to show here. That's why. Thank you. Here you are showing the result for one spectrogram. Yeah. 10 seconds. And what are the results for others? And I imagine you have done repeated methods for other spectrum. So actually, here what we do to simplify is we take the spectrograph of one of the array that is in the middle of the scene, because it's the one that hears the most of the sources, because Alitopstetricans are closer to Z2. That's why we did that. But exactly. If you look at the Z4, for example, it will not be exactly the same spectrogram, and we will not have as much detection, for example. Yeah, so that's what we did, but it's not, maybe it's not the best method to do. I don't know. Do you have an idea why there is one array that is not working? It's not that it's not working, it's just that the individuals are really closer to Z2, and Alits are emitting calls that are not that loud. So obviously it's better seen with this one.
I think that you said that for one individual, for example, you can have 10 points within different position. Like,
yeah.
For example, one individual that is far from the
yeah.
You have maybe 5 positions that are
in a cluster of points.
Yeah, you can have many positions for one color.
Yeah. Yes.
The combination with that. But let's say just to erase all the and just.
Yeah. Yeah, we did that kind of thing on spherical harmonic beamforming. It's called the filter max RE, and so it's clearing the map. So and it doesn't change anything because we look at the maximum points on the map. So it actually doesn't change the results. Yeah.
I have a question regarding the when you measure the time difference arrival between you clapped 15 times. What?
Yeah. Well, if you want to have good results, you have to have like it's. I had to do more than 5 claps for my number of arrays of microphones and their positions. So I did 14 to be sure that we have. Actually, the more you have sources, the most accurate will be the positions.
15 claps in different positions?
Yeah.
Okay. As of saying, have you tried comparing when the heights are the same to the situation when the heights are different?
The height of the arrays,
So
you mean? Yeah. At first, in the anechoic chamber, we did an experiment with the same heights, but actually it's worse because if you have different heights, you can have a more precise 3D position in 3D.
And in the lab experiment, the target was inside inside the.
It was in the semi-anechoic room.
Yeah, I mean the speaker, for example, when the array was all around the speaker, because here I think the sources are.
We did both. We put sources inside and outside the network.
And there is difference?
Well, yeah. If you're inside, you actually you see better. You have a better position because like you triangulate in a better way. And as I said here, the DOAs are more different for each array.
You said that you couldn't compare inside the top.
Yeah.
Think of using a tripod or perch in order to be inside or kind of way?
Yeah, well, we thought of different stuffs, but actually it's difficult. We don't have the material here. Also, we could have used the GPS, little GPS for every array, but we don't have it. Like, we used what we had and tried to do the best with that.
Regarding the validation, is that a study to made when this type of frogs does group depending on their frequency? Because it seems like the three of, let's say there are three at the high pitch, they seem to be close together, whereas the low frequency one is kind of apart. And in more general way for the validation, you intend to like I don't know, shoot some cameras to try to see exactly where the frogs are during your measurement? Or do you intend to do some kind of?
Actually, I think they are of different age. They probably of different age.
The frogs?
Yeah, the different tonal range corresponded to different age of frog.
Actually, each frog during most of his life is having the same frequency call.
Since you were born.
Maybe not born, but yeah. So we thought of it. The problem is that you really don't see it. Like there are really high grass. You can't see it. It's at night. What? They can be inside the rock also. Yeah. And they are like this size. But here we couldn't see anything. It was impossible. So it would be nice to have cameras and videos, but we can't. We don't have it.
Yeah,
and so actually there haven't been any studies on that, on the spatial. So that's why we do that. And for now, yeah, we don't have validation, proper validations of that visually. Yeah.
Yeah, it's also interesting to ask why we want to detect their place. We don't want to approach them. We don't want to approach them. We need to leave them live peacefully So why you are detecting
The positions?
Yeah.
We want to know the positions because we want to know what is the spatio-temporal pattern, if there's a pattern in choruses. We don't know yet. We know that there are a lot of patterns in time, but in the spatial, spatially we don't know. That's why we do that. We want to know if maybe because actually females are attracted by the lowest voice. So maybe the lowest voice caller is at one specific place and the others are around, or we don't know, and we want to check that.
We can say that monitoring of time, space, position, different sources is a scientific task. But you have mentioned at the beginning another task is the fight for the survival of endangered species. So how in general such monitoring would fit into these objectives regarding the because at the first sight one would say that if you allow monitoring of the condition it will fight these species, not want to help them. So what is the general framework? Scientific techniques identifying position and let's say this final goal.
Well, I would say that it's helping to know a bit better those kind of species, and for now, bioacoustic studies are really focused on like birds or species that maybe are more visually pleasant for human. And as I said, anurans are endangered because of climate change. So I think it's a good thing to study what's happening in their populations and like to get to know better the individuals and species of anurans. And if you, I think if you propagate like how they live, what they do, you will, if you know better a species, you will want to don't want to say protect, but you will more want to be aware of what's happening for them. So, for example, I went to with LPO, Ligue pour les oiseaux. I went out to listen if there are some alite obstetricans in Mont d'Or. And so we heard that there are a lot in people's habitations in their garden, and they maybe don't know it actually because we are not used to that kind of calls. And so what they do is to go to people's house, knock at the door, and try to tell them, yeah, you have an endangered species in your garden. Be careful, don't kill them. Yeah, kind of thing.
You can take one last question for Madame Lucienne.
Okay, so I see a green signal around 1,300 is like PV.
Yeah.
So what if you compute the spectrum of that signal in order to study the characteristic frequency? So the characteristic frequency can then be used to estimate the intensity of the animal, let's say for specific or for example. So is it possible to
extract the signal of one?
yes,
yeah,
because some of the signal are fluid emissions using the spectrum.
Yeah. Well, actually, what we know is that one individual is emitting at the same frequency, the same intensity. So normally one array will see the same way one individual that calls several times. We also know that the distance between the temporal distance between like the timing is really precise, and one species will call every half second, for example, and it will be the same all the time, except when they stop calling and then they call again. So yeah, we can extract that and extract like the intensity of the call and try to know.
Which type of frog, if it's possible,
You mean tell which which frog we
type of frog we always like you see characteristic.
Yeah.
We should just be confident whether it's not loud and then condition condition like frog usually changes between maybe two different seasons. Like maybe in summer they are loud and in winter they're not loud, but the characteristic is basically really possible. So I think that information for a crucial role to maybe know which type of frog it's. Yeah, maybe
Yeah.
if you want to.
So actually, we know that if you have a change of humidity or temperature, the call will not be exactly the same. So if you go one day and then the next day, maybe it will change slightly, and you will not be able to say, okay, it's the exact same call for the exact same individual. That we know that it changed slightly, but in like a few minutes, it will not change. It will be the same. That's why, like we have a lot of informations. We have a lot of signals. So here we know that in those minutes it will not change. I don't know if I answered to the question actually.
Louise Chiocchetti, thank you again for the seminar.
Thank you.
And thank you everyone for joining this seminar here at the LVA or online. And, if you want to present the seminar as well, feel free to contact me or Louise. And we will be very glad to